Okay, are there any questions before we begin?
Nope.
Thank you. Oh, I don't have the summer on there yet. Well, I won't be able to show you what we're going to cover today, but I'll tell you that we're going to cover chapter 1 and chapter 10. And we probably won't finish today, but hopefully we'll get close to finishing. Oh, come on. Are you there? I'm having a hard time. There we go. Get in the window. There we go. So you should be able to see the screen now. Let's see the help. All right, so we talked about this slide last time. Microorganisms and disease. As I stated, we're going to cover chapter 1 and chapter 10 today. Let me remind you that the lab assignment 00, the worksheet for that lab assignment must be turned in by Friday at 8 p.m. I'll use that assignment to determine that you guys have registered for the class and that you are attending. If you do not turn in the worksheet, you will be dropped from the class. Any questions about any of that? The other assignments must be due at the normal time, and that's Saturday at 11:59 p.m. That includes the worksheet for the lecture, which covers chapter 1, and then the lab worksheet that we'll do today, and that's lab 01. And we haven't done that yet, and I'll talk about that in the lab today. All right, we talked about microorganisms and we mentioned they're small. How small are they? Anyone know? Microorganisms are small enough that we cannot see them with our naked eye. Most bacteria are around 1 micron. That's 1 micrometer in diameter. And this might be E. coli. It's a bacteria in our gut. And if you can see this, let me blow that up a little. It's about 1 micrometer in length, a little bit less than a micrometer in width. And of course, that's much longer than 1 micrometer. But E. coli is a little bit larger than your average bacteria. They do vary in size. As you can see, this is a mold, a mold fungus, and this one is 15 micrometers long. This is also considered a microbe. This is actually a white blood cell, and it's much larger than bacteria. And there we see it's 10 micrometers, so this would be well, at least 50 micrometers in length, maybe as high as 100 micrometers. Here is a microbe, and it's actually, you're not seeing the cells. In fact, each of these dots is a cell. And this is a colonial microbe. And this would be the parent colony. And these little colonies inside of it are baby colonies. And so each of these dots is a cell of the baby colony. And this is Volvox. It'll open up and then that'll release the baby colonies to go out and colonize different areas. I think that's a virus on a CD4 cell. Yeah, that's HIV, which is right here. And viruses are very small. This right here is 50 micrometers. We did mention that microbes play beneficial roles in the environment as well as beneficial roles for us. Some examples of the beneficial roles that microorganisms play is that they're very good at decomposing organic waste. All organic waste is either broken down by a microbe or a fungi. And mushrooms are fungi and they do decompose, but that would be the largest decomposer. Microbes are also producers in the ecosystem. We have producers which engage in photosynthesis, as well as producers which engage in chemosynthesis. Producers are organisms which make their own food, usually from sunlight, and that would be a photosynthetic producer. But there are producers which use chemosynthesis to make their own food, where they can make sugar from carbon dioxide and water. Producers are involved in the nitrogen cycle, and they are, not producers, microbes are involved in the nitrogen cycle, and they're what's running the nitrogen cycle. Microbes produce many substances for us, for humans, such as industrial chemicals, examples being ethanol and acetone. We use them to make compounds for us. And that would be like the gasoline you get in your, not the ethanol you get in your gasoline. That's made by microbes. We use them to make certain insecticides, and we'll talk a little bit about that in a little bit, where we can use microbes to make chemical insecticides, and they're less harmful insecticides than the man-made insecticides. We use microbes to ferment food for us, and this has been used for thousands or millions of years. We use microbes to make vinegar for us. We use microbes to make cheese for us. We use microbes to make bread for us. In fact, if you put yeast in bread, that is a microbe. And what we're talking about is here is also fermented bread, which yeast does ferment, but I'm talking about sourdough bread. And sourdough bread uses both yeast and bacteria to make the bread. And then we use microbes to make various substances like beer and wine. We use microbes to make products that are used in manufacturing. One example is cellulase, which breaks down cellulose. We use microbes to make antibiotics, like penicillin G is made by a microbe. And then many other products that are used in medicine. Like today, human insulin is made by microbes. We've taken the human gene for insulin and put it into a bacteria, and then we grow up the insulin and the bacteria. Any question about any of that? All right. So modern biotechnology uses microbes, and so does genetic engineering. Biotechnology is the use of microbes and organisms to produce foods and other products for human use. Biotechnology is not something new. It's actually centuries old. Like when we've been fermenting cheese, making, I don't know, cheddar cheese, that's at least centuries old, okay? The new aspect of biotechnology, which we call genetic engineering, is a fairly new technique of biotechnology where we use microbes to make things for us, such as some applications include the variety of, we make proteins from bacteria and fungi. An example of that would be where we make vaccines and various enzymes in bacteria and fungi. Another application is gene therapy, where we replace missing or defective genes. Like a child who's born with an immune defect, they're missing the correct gene to make their immune system function fully. And we can get that gene out and put it in their cells using biotechnology or genetic engineering. And usually we put it in a virus and then infect their cells and then put in the cells back into them. And then their immune system will work. The only trouble is that this is only a temporary fix, and then the child must have this gene therapy every few months, but it does allow their immune system to function normally. And then there's genetically modified bacteria, which we can use to protect crops from insect damage or from freezing. Another example, if you remember the flavor saver tomato, genetically modified tomato. Supposedly it didn't rot as quickly on the store, and so it would keep longer. And I don't see the flavor saver tomato anymore in the store. So I'm guessing it wasn't something that a lot of people bought. And so you need to look very hard to find it now. Microbes are important for human welfare. Microbes are important in our microbial ecology. Microbial ecology is where microbes interact in the ecosystem. We use microbial ecology for bacteria to recycle many nutrients, such as carbon, nitrogen, I've already talked about. Nutrients, if something dies in the environment, it's microbes that largely break it down. It is true that mushrooms and other fungi can break it down, but most fungi are microbes, not mushrooms. But anyways, what happens is the microbes break it down and then make those nutrients available for the ecosystem. Meaning they make the nutrients recycle in the ecosystem. Microbes are important in the sulfur recycling in the ecosystem, as well as phosphorus recycling in the ecosystem. And then these nutrients, carbon, nitrogen, sulfur, phosphorus, and other nutrients can then be used by other plants and animals. The carbon, the nitrogen, the sulfur, and the nutrient recycling in the ecosystem are examples of microbial mediated biochemical cycles that help support life on Earth. Without the microbes, these nutrients would not recycle, and eventually they would be used up in the ecosystem. And then there'd be no more of these nutrients available for life. So the microbes are very important for supporting life on the planet Earth. The microbial mediated biochemical cycles were first studied by Winogradsky and Bajering. And they found these microbial mediated biochemical cycles because they were studying them, and they were studying microbes in batch, meaning they didn't study microbe by only studying one species. They studied lots of microbes living together in the ecology. Anyways, they were the first to show that the recycling of nutrients, such as nitrogen and sulfur, happen by microbial mediated biochemical cycles. Any question about any of that? Microbes are also important to human welfare, and when we have a problem, we can use microbes to help remedy the problem. And this is bioremediation. It literally means life, and to remedy, where we use living organisms to deal with an environmental problem. Like these men are spraying microbes on this oil spill for the microbes to break down the oil, the pollution that is oil. And we can use microbes to break down sewage and other pollutants, such as mercury. Now, bacteria do not totally break down mercury into its other elements, because mercury is actually an element, but it can convert mercury into a more biologically friendly compound. But oil is broken down into carbon dioxide and water. And so it would be totally broken down in the ecosystem, where we use microbes to degrade and to detoxify pollutants, breaking them down to less harmful substances. All right, any question about any of that? Microbes also make biological insecticides, and these microbes are pathogenic to the insects, but they are not pathogenic to humans or to animals. In fact, they're harmless to humans and other animals and plants. An example of that is Bacillus thuringiensis, which this is a bacteria, and it's fatal to many insects, but harmless to humans. And we can use it to prevent crop damage from insects, as well as prevent disease transmission spreading among our crops. And my father actually used Bacillus thuringiensis back in the 1960s. So this has been used and known about for many years. And he put it on the plants to prevent the apples from getting worms. And it did work. However, you do need to apply it every 2 weeks, which actually, when you're spraying to keep the apples free of worms, you need to spray about every 2 weeks. So it's not more common, but you do also need to replace it after the rain. And the trouble was, he was in Western Oregon. And as you know, we get frequent rain when the apples are first getting started in the spring, as well as in the fall. And so you do need to replace it after the rain, and he didn't do that. And so the apples did have some worms, but they were less wormy than they would have been if you didn't use it. Any question about any of that? All right, for the apples, like I said, you need to replace it about every 2 weeks. And I'm not sure why the Bacillus thuringiensis comes off of the apples, if that's because of the wind blowing it off, or if it's because you always get rain in about 2 weeks. I don't know. But it does need to be applied about every 2 weeks, and then whenever the rain occurs. All right, any questions about how microbes are important to human welfare as well as the ecosystem? If not, we're going to change topics a little bit and go into the naming and classifying of microorganisms. And it is true that we're going to talk specifically about microorganisms, but this does apply to the naming and classifying of all organisms. Scientific names is covered in chapter 1 and chapter 10. It was first begun in the 18th century in 1735 by this individual here who established the system for scientific nomenclature. Does anybody know his name? Anyone know who this is? You should have had this in biology. Is it 160, your previous course? Okay, I'm not hearing anyone say anything. This is Carolus Linnaeus, and he was so fond of Latin that he Latinized his name. It actually was something like Carl Linné, and he changed it to Carolus Linnaeus to make it Latin. What he did was he established the scientific nomenclature where he used two names to name each organism. So each species has two names, a genus name and a specific epithet name. So you can't use sapiens to name the species because that's a specific epithet name. For humans, you need to call them Homo sapiens, Homo being the genus, and then Homo sapien being the species. This is called the scientific nomenclature, as well as the binomial nomenclature, because bi meaning two is binomial, meaning two names to name each species. And it was such a good system, it's still being used today, meaning scientists continue to use his system, the binomial nomenclature that he came down with. He also started the classifying of microorganisms, putting them into groups, are ever larger and larger groups, meaning similar animals are put into, well, a genus, and then they're put into a family, and then eventually into other larger groups, the kingdom and the domain. We don't use his classifying anymore because he grouped everything into two kingdoms, animals and plants. And as you know, there are more kingdoms today than two kingdoms. But he started it. And for example, if you put Homo sapiens into the genus Homo, and that we do continue. I guess he would group humans into animals, but the largest grouping, the domain, was not known in his day. All right, let's go back to the nomenclature system. The names are italicized or underlined. So like if I were to write it on the board, I would underline it, meaning that it is a scientific name. Italicized, I would type. The genus name is always capitalized, and the specific epithet name is always lowercase. All names are Latinized, and they're used worldwide, meaning whatever language you use, wherever you are in the world, we would use the same name to name the species. Like Homo sapiens is used by everyone to discuss species, the human species. Staphylococcus aureus is used throughout the world, whatever language you're using, to describe Staphylococcus aureus, that bacteria. The name may be descriptive or it may honor a scientist. Homo sapien is an example, and this literally means wise man who walks on 2 feet, Homo sapien. Escherichia coli is both descriptive and honoring a scientist. Esther was an early microbiologist, and Escherichia honors him. And then it's descriptive because E. coli lives in the gut, and coli means colon, meaning lower intestine, and that's where E. coli lives. Staphylococcus aureus is descriptive. It literally means golden cells that are shaped like a ball. That's what a coccus is. And grows like a grape. That's what staphylo means, meaning a cluster of grapes. Any question about any of that? After first use, scientific names may be abbreviated by using the first letter of the genus, and it will be capitalized, and the specific epithet name. So Staphylococcus aureus is what we use the first time we discuss the name, but all other uses of the name may be abbreviated as S. aureus. And Escherichia coli should be used the first time. And then E. coli is used all other times. However, E. coli is so well known and so well used that even among scientists, they frequently will just say E. coli. And we all know what you mean by that. But specifically, when you're using the nomenclature system, it should be spelled out the first time, Escherichia coli, and then all other times use it as E. coli. Any question about any of that? By the way, Staphylococcus aureus can be found on the skin. About 20% of Americans have Staphylococcus aureus on their skin, and it's actually a human pathogen causing skin disease. So if you're somebody who frequently gets boils, you might have Staphylococcus aureus living on your skin. E. coli is found in the human body on the large intestine and the colon. Any question about any of that? All right. So when we're classifying microorganisms, grouping them into larger and larger groups, we do need to look at the classification of all living organisms on the planet Earth before we talk about the classifying of microorganisms. The first group of all living organisms that we split a living organism into follows the 3-domain system, meaning the first group that we group things into is the domain, and there are 3 of them. So here we're looking at the 3 domains, and sometimes this is called the tree of life because the 3 domains branch like a 3-branched tree of life. And then the domain branches into smaller and smaller grouping, meaning that the tree of life branches like branches on a tree. The first domain has 3 domains, and the first is bacteria, the second being domain archaea, sometimes called archaebacteria, and then the third domain is eukarya. The domains are defined and based on the genetic data of the species, meaning we can sequence the DNA of a species and then determine which domain it belongs to, because all species in one domain will have their DNA more similar in the sequence of that DNA to other members in the domain compared to a member that is not in the domain. Any question about any of that? So the domain is defined, and what you do to determine which domain you go to is you simply look at the sequence of DNA of that species. And all members of bacteria have a similar sequence of similar sequence of DNA, and it's much more similar to any member in the bacteria than it is to any member that is not in domain bacteria. Now, initially, when the microbiologists were studying the microbes, we did not know that there were 2 domains of prokaryotes, meaning domain bacteria and archaea. So this came as a surprise when people started sequencing the DNA of species back in the 1970s, that there were 3 domains, bacteria, archaea, and eukarya. We assumed that the bacteria, the prokaryotes, that they were all single cells. So we assumed that they were similar to each other. But actually the archaea in their sequence of DNA is very different from the sequence of DNA in the bacteria. And actually the sequence of archaea is a little more similar to the sequence of eukarya, just a little bit, than it is to the domain bacteria. And that's because if you look very closely right here, in the tree of life, the archaea and the eukarya were on the same branch for a little bit, just a little bit, before they separated into 2 branches. And so the archaea are actually a little more similar to the DNA sequence of the eukarya than they are to the bacteria. And that's just a slight difference, meaning all the archaea are similar to each other, but they're very different from the bacteria and the eukarya. Any questions about any of that? All right. After the grouping of the domains, we then have the grouping of the kingdom, and the domain eukarya breaks down into 4 kingdoms: kingdom plant or plantae, kingdom animalia, the animals, kingdom fungi, and then kingdom protista. Any question about that? Now, for our system, we're going to say the bacteria all belong to kingdom bacteria, and the domain archaea break down into the domain archaea or the kingdom archaea, so that there are 6 kingdoms: kingdom bacteria, kingdom archaea, kingdom plants, kingdom animals, kingdom fungi, kingdom protista. Any question about any of that? All right, no questions. However, you should know that the kingdom is not defined. There is no definition of what a kingdom is. And so there is arguments about which kingdom a species belongs to. For example, some people argue that the kelp, the very large seaweed which grows in the Oregon and Washington coast, washes up on the Oregon and Washington beaches. Some people argue that it belongs into kingdom plants, and other people argue it belongs into kingdom Protista. And there is no definition for what a kingdom is, so I guess you're allowed to argue that. Anyways, there are arguments as to what species belongs into what kingdom, because the kingdom is not defined. And the number of kingdoms is also up for debate, meaning among scientists, different scientists use different kingdom numbers. Some people use only the 3 kingdom approach, and that would be each domain is 1 kingdom, and that would be okay because they're just, the domains is the kingdom. We're not going to use that approach. Some people use the 5 kingdom approach, and we're not going to talk about what that is, but there's 4 kingdoms: plant, animals, fungi, and protista. And then they lump the prokaryotes together. And I consider that a mistake because the bacteria should not be in the same kingdom as the archaea because they're in different domains. And the domain is larger than a kingdom. And then people use the 6 kingdom approach, which we're going to use. Kingdom bacteria, kingdom archaea, kingdom plants, kingdom animals, kingdom fungi, kingdom protista. The problem with the 6 kingdom approach is that protista is defined as the kingdom that is a eukaryote, that is not a plant, not an animal, not a fungi. So the kingdom Protista, by definition, is not a monophyletic group. Any questions about that? And I guess the kingdom Protista has members in it which are very different from each other in their sequence of DNA as well as in their biochemical makeup. Other people say there are 9 kingdoms, and what they do is they divide the kingdom Protista into different kingdoms. People say, no, there's not 9 kingdoms, there's 15 kingdoms. And once again, they divide the kingdom Protista into ever more groups. And this might be the 15 kingdom approach where we have each of these groups here as a kingdom. And then some people have the 20 kingdom approach where you divide the Protista into different kingdoms. You divide the plants into at least 2 kingdoms. And then you start dividing the archaea and the bacteria into different kingdoms. And I think there's 3 kingdoms of the archaea, which are shown here. I don't know how many kingdoms there are in the bacteria, but there's at least 3 in the archaea. And then you can divide the kingdoms into higher numbers than 20. And I don't know the highest number that people use. Any question about that? So the kingdom, we're going to use the 6 kingdom approach, but I'm not going to tell you that this is the only approach that's used. And the number of kingdoms is actually being debated by scientists. And it's been in debate for more than 20 years, so I don't think it's ever going to be solved. And there is no definition for what a kingdom is, so scientists also argue about what kingdom a species belongs to. You can break down the kingdoms. We're not going to go into that. Let me see. Repeaters. So into the phyla would be next. We're not going to break down into the smaller groups. All the way down to species, and then you even have subspecies or what do you call that? Races. All right, so let's talk about the three domain system a little more. It is defined. There are three domains: bacteria, archaea, and eukarya. The textbook talks about prokaryote cells and eukaryote cells as a cell type. So a prokaryote is a cell type. And the prokaryotic cell type is found in two domains, the bacteria as well as the domain archaea. And then the second cell type, eukaryotes, is found only in the domain eukarya, the eukaryotes. These are fundamentally different, the cell type. A prokaryote cell doesn't have a nucleus, doesn't have membrane-bound organelles, and chemically it's a little more simpler and structurally a more simpler cell than a eukaryote cell, which does have a nucleus and membrane-bound organelles. Now, the cell type may be called the superkingdom. And the superkingdom is another way that people group organisms. And so you first group organisms into the three domain system, and then you break it down into the superkingdom. We're not going to use that, but if ever you hear the word superkingdom, it's been around much longer than the domain system. And there are two superkingdoms, the prokaryotes and the eukaryotes. Any question about that? The superkingdom is defined, by the way. Any cell that has a nucleus belongs to the eukaryotes, and any cell that does not belongs to the prokaryotes. And I do remember they did find a prokaryotic cell. You don't need to know this, which there's one species that actually has a primitive nucleus, and they have not changed the definition of a eukaryote, but so far there's only one species where there's an exception to this, and we're just using it as an exception. And I'm sorry I even brought it up because I'm sure you've never heard about it. But the superkingdom is pretty much defined, so there is no argument about what species belongs to what superkingdom. The way there is arguments about what species belongs to a kingdom. All right, let's talk a little bit more about the domain bacteria. It has a cell type, prokaryotes. In the cell wall of bacteria, we find peptidoglycan. And peptidoglycan is the main component of the cell wall. Bacteria multiply mostly by binary fission, which means the cell literally divides into two. And we'll talk a little bit more about binary fission in a later lesson. For any energy use, the members of domain bacteria can use any energy source. They can use an organic chemical, which is what we do. I don't know if you can see this. There's a piece of food. We get our energy need from an organic chemical like glucose. But there are members of the domain bacteria which can get their energy need from an inorganic chemical like hydrogen sulfide or sulfate or iron compound. They're using energy from an inorganic chemical. These are the chemosynthetic bacteria. We'll talk a little bit more about them later. And then there are the third way you can get energy is from light, and the photosynthetic organisms get their energy needs from light. Most of the members of domain bacteria are either free-living decomposers or primary producers. So most of the members of the domain bacteria are decomposers, meaning free-living. They don't live on and off of something to decompose the person, for example, or a primary producer. But there are some members of the domain bacteria which are parasitic, meaning they're living off of another organism, doing harm to that organism. And then some members of the domain bacteria are pathogenic, meaning they are capable of causing disease. Any question about any of that? All right. The next domain we want to talk about is the domain Archaea. These have prokaryotic cell types. Does anybody know, is peptidoglycan found in the Archaea? Is peptidoglycan found in the Archaea? If you guess, you got a 50% chance of an answer. Nobody's gonna guess?
I'll say yes.
You wanna guess again?
No.
Yeah, the answer is no. Peptidoglycan is generally not found in the cell walls of Archaea, and it is not found in the Archaea or in the domain Eukarya. So peptidoglycan is only found in the domain bacteria. The Archaea, some of them are found in the extreme environments, and that is where we initially found the Archaea. They include the methanogens, those bacteria that make methane. They include the extreme halophiles. Halophile means salt lover, and these are organisms that live in high salt concentration. There are extreme thermophiles, which are Archaea, and they live at very high temperatures. However, if we look at the biomes, we can find the Archaea in all of the Earth's biomes. So the Archaea are found everywhere on the planet Earth in all of the biomes. They were initially found in the extreme environments, and some authors say that they are found in the extreme environments. That's actually, gosh, it's at least 15 years old, but they are found everywhere. They make up a significant number of the organisms found in the soil, as well as in seaweed, meaning we find a significant number of the microbes in the ocean, which are Archaea. So they're found everywhere in every biome. They are not just extreme, found in extreme environments. Does anybody know, are the Archaea pathogenic? Are any members of the Archaea capable of causing human disease? Nobody wants to guess? None of the Archaea that we know of are pathogenic, meaning no human disease that we know of is caused by Archaea. For this reason, and this reason alone, we are not going to talk much about the Archaea after this lesson. And that's not that the archaea are not important. Obviously, they make up a significant percentage of the living organisms on the planet. But we're not going to talk about them because the focus of this class is the microbes that are capable of causing human disease. And so we're going to talk largely about pathogenic organisms. And guess what? Those are all bacteria or animals, meaning the parasitic worms are animals. And so all of the infectious diseases are caused by either bacteria or by the parasitic worms. And of course, there are pathogens which are not cells, the viruses and the prions. But of all living organisms, only the bacteria causes human disease. So only they are pathogenic. Any question about any of that? All right, let's talk a little bit about the domain eukarya. We're going to split the domain eukarya into 4 eukaryotic kingdoms: the kingdom plants, the kingdom animals, the kingdom fungi, and the kingdom protista. Realize that the kingdom protista is just a collection of all the eukaryotes, which are not plants, animals, or fungi. So it's not monophyletic. None of the members of the kingdom eukaryote have peptidoglycan. So all members of eukarya lack peptidoglycan. Peptidoglycan is only found in the cell walls of bacteria. Excuse me. Let's talk a little bit about the kingdom fungi. You don't need to know, but their cell walls are largely made of chitin. You don't need to know that, but they do use organic chemicals for their energy. And that's the same with animals. We use for our energy source an organic chemical such as sugar. Here we're looking at a multicellular mold or a mushroom, which is multicellular, consisting of masses of mycelia composed of filaments that we call hyphae. So right there, that would be a hyphae, one cell. The kingdom fungi also includes some, what's the word I'm looking for? Some microbes, and that would be the unicellular yeast, as well as the unicellular molds. Most members of, actually all members of the kingdom fungi are either decomposers or a pathogen. So they either decompose and break down something like yeast or a mold or even a mushroom. It's a decomposer. Or they could be a pathogen like, I forgot the name of that. A yeast that grows on the human skin and causes various diseases like nail, no, not nail rot or nail fungal disease. I'm forgetting the name of it. Sorry. It'll come to me. Anyways, some fungi are pathogens causing human disease, but not most. Most are decomposers. Kingdom protista is a grouping of eukaryotes which do not belong to animals, plants, or fungi. And we can break down the protista into the plant-like protists, and that include the some of the algae, if they're single-celled algae. They have cellulose in their cell walls, meaning similar to plants. They engage in photosynthesis for obtaining their energy, and they produce molecular oxygen as well as organic compounds, meaning they make glucose and all of their food from CO2 and water. Anyways, those are the plant-like protists, which are mainly the algae. The animal-like protists we call the protozoa. That include the amoeba, paramecium, and the euglena. They absorb or ingest organic chemicals to obtain their food. So they do not make their food from photosynthesis or chemosynthesis. They have to ingest or absorb their food, same as we do. They may be motile, and they can be motile by three things: by pseudopods, shown here. And this organism, if it's moving this way, would put out cytoplasm right here, and then pull in the cytoplasm there. And this part here or that part there is a pseudopod. They can also be motile by a cilia, which is a hair-like structure which beats, propelling the organism to move in the opposite direction of the beat. And there's usually lots of cilia on a organism. The flagella is the third way that a eukaryote can move, and it's a whip-like structure. There's usually one or a few flagella on a cell. Sperm is a human cell which can move, and it has a flagella. I think there are only two cells in humans that have flagella, and that's the sperm and the cells in the throat. The cells in the throat move the mucus. It doesn't move the wait a minute, that's not flagellum, that's cilia. Is that right? Let me think about that. Yeah, that's cilia. So I think sperm is the only cell that has a flagella. Human cells can move also by pseudopods, like white blood cells move by white blood cells. So they can move by more than just flagella. All right, protozoa, some of them are parasites, living on and off another organism. And we'll talk a little bit about these. The parasitic protozoa causing human disease, like, sorry, I'm drawing a blank on what this is called. Malaria, caused by a parasite, which is a protozoa. And then the third grouping of the kingdom protists are the fungus-like protists. And we won't talk much about them, but water molds, which cause potato blight, which cause the potatoes to rot. In Ireland, which caused a lot of the Irish to come over to America, was actually caused by the fungus-like protists. Any question about any of that? All right, kingdom plantae, we're not going to talk much about because they do not cause human disease. We can separate the kingdom plantae into the mosses, the ferns, the conifers, and the flowering plants. All members of the plants are multicellular. So all plants are multicellular. None of them are a microbe. Any question about any of that? We're not going to study them, like I said, because they don't cause human disease. Of the kingdom animal, we can separate them into the sponges, the worms, the scientific name being helminth, the insects, and then the vertebrates. All of the animals are also multicellular. So none of the animals are microbes. Any question about that? We will study a few of the animals because some of the helminths do cause human disease. Okay? Any question about that? All right. So when we're talking about microorganisms, what do we mean by what comes under the umbrella of a microorganism? What belongs to a microorganism? Well, the bacteria are all microorganisms. The archaea are all microorganisms. Viruses and prions are also microbes. I don't want to say microorganisms because a virus and a prion is not an organism. They do not have cells, but they are a microbe and they do cause human disease. So they do fall under the umbrella of what is a microorganism. Some of the eukarya are also a microorganism. All of the members of protozoa are a microbe. Some of the molds and some of the yeast, actually all of the yeast, are microbes. Okay? If they're single celled, they are a microbe. Some of the algae are also microbes. If they're single celled algae, they are a microbe. There are a few of the multi-celled algae, which are also microbes, and you don't need to know that, like Volvox. To see it, you need a microscope, and it has, I don't know how many cells, but it doesn't have a large number of cells. And so it is a microbe, okay? It's the only example of a microbe that I know of, which is multicellular. The helminths fall under the microorganisms. And the helminths, remember, are the worms, but most of the worms aren't microscopic, are they? Do you need a microscope to see most of the parasitic worms? That's a yes or no. Oh, you guys really don't like to contribute. Most of the worms are not microscopic. Most of the adult worms are large enough that we can see them with the human eye. The largest, I don't remember the name of that. The beef tapeworm is meters long. Let me see, is it something like 20, up to 20 meters long or something like that? So it's very long, is the point. However, there are certain life stages of the parasitic worms, which are microscopic. And it's the microscopic version of the worm, which is usually the infective vehicle for infecting a human, meaning most humans get a parasitic worm by consuming the fertilized egg of the worm. And so some life stages of the parasitic worms are microorganisms. For this reason, we consider the helminths to be a microorganism. And then the other reason is that some of the helminths cause human disease, and organisms which cause human disease have been studied by microbiologists. And so microbiologists consider the helminths to be a microorganism. And in all of them, there are various life stages which are microscopic. Do you realize the helminths are, as an adult, a multicellular animal? Although many are parasitic, a parasitic flatworm or a parasitic roundworm, they are not strictly microorganisms. Usually the adult life stage of the parasitic worm is not microscopic. Only some of the life cycle is microscopic. You should realize that although we're going to talk about the parasitic worms, that the helminths, the parasitic worms, or the worms, not all of them are parasitic. Most of the helminths are not parasitic. Most of them are free-living worms, do not cause disease. In fact, there's one phyla of the parasitic worms, the annelids, which none of the members are parasites. An annelid that you know is the earthworm, the worms that crawl in the soil. That's an annelid. It doesn't cause human disease. Only some of the members of the roundworms, and I think all of the members of the, no, not all the members of the flatworms. Some of the members of the flatworms cause disease, and these are the parasitic flatworms and roundworms. But not all members of the flatworms and roundworms are. Most of them are free living and they do not cause disease. Any question about any of that? All right, before I go further, let me mention that although we group the helminths together as a worm, because they look similar to human eyes, this is actually a misnomer. And because the flatworms and the roundworms are as different from each other as they are to humans. So when we sequence a flatworm and we sequence a roundworm, the DNA sequence will differ from each other as different as they are to a human. So these flatworms and roundworms are not in the same group, scientifically speaking. They are as different from each other as they are different from humans. Any question about any of that? All right. So let's talk a little bit about viruses. That's from a previous lesson. As we stated, viruses are acellular. They do have a genome, which can be either DNA or RNA. And it is the core molecule of the virus, meaning the innermost molecule of the virus. But a virus never has both DNA and RNA. It's either DNA or RNA. And it's a little unusual for a cell to have RNA as its genome. All cells have DNA for their genome. But for viruses, we do have, not some, at least half of them, which have RNA for its genome. All viruses have their core molecule, meaning the DNA or RNA, surrounded by a protein coat. The protein coat may be enclosed inside a lipid envelope. So the virus may have an envelope, a lipid envelope, but viruses do not have to have a lipid envelope. Many of them do not have a lipid envelope, in which case all they have is the protein coat and the genome. And viruses only replicate when they infect their host cell. Viruses cannot replicate on their own. So if they're outside their host cell, they do not replicate. In fact, when they're outside their host cell, viruses are metabolically inert. They're only metabolic when they're inside their host cell. Viruses are not part of the three-domain system because they do not have cells. They are acellular. Any question about any of that? All right, let's talk a little bit about the three domains. The domain bacteria has cell type prokaryote. The domain archaea has cell type prokaryote, but the domain eukarya or the eukaryotes has the cell type eukaryote. When we look at the chromosome, the domain bacteria has one chromosome which is circular, meaning it's in a circle. The place where it ends is the place where it begins. There are no histones on the chromosome. It's true there are proteins on the chromosome, but there's no histone proteins on the chromosome of the domain bacteria. There are also no introns, and we'll talk about introns much later in the term. An intron is a region of the gene which specifically codes for the protein or the gene product. The domain archaea generally has one chromosome, which is also circular. You don't need to know this, but it does have histones on that chromosome. And you don't need to know this, but it has introns on the chromosome or in the gene. The domain eukaryotes generally has many chromosomes, like humans have 46 chromosomes, and they are linear chromosomes. Like when we look at the X chromosome, it has a beginning place and an ending place. So it's a linear chromosome. It's not in a circle. The eukaryotes, you should know this, have histone proteins on the chromosome. In fact, the chromosome is more than just DNA. I don't know what percentage is, but it's close to 50% of the human chromosome is protein. And most of that protein is the histone protein. And of course, there are introns in the eukaryotic DNA or the eukaryotic chromosome. When we look at the ribosomes, the domain bacteria have small ribosomes that are 70x. The archaea have also the same size ribosome, 70s. The size, you don't really need to know this, but it stands for the sedimentation coefficient, where the ribosome sediments in an ultracentrifuge gradient. And that's why it's an S, the sedimentation coefficient. And the point is that the bacteria and archaea ribosomes are smaller than the eukaryote ribosome, which is larger, and it sediments at 80S, a place lower down in the ultracentrifuge gradient. Any question about that? All right, we've already mentioned peptidoglycan is found in the cell wall of domain bacteria, and that's the only place it's found. Not found generally in the archaea and not found in the eukaryotes. Any question about that? All right. If there's no questions about the different domains, I'm going to switch topics and start talking about the history of microbiology. This is a really interesting part of the chapter, and we're only going to briefly talk about the history of microbiology, going over the main discoveries, who made the discovery. I'll mention the year, but you do not need to know the year, except for the very first year, which I'll talk about, okay? So you should know the brief history of microbiology, but you don't need to know great detail and specifics, but you should know who discovered what, and then you should know generally a little bit about what. You don't need to know great details, but you should know specifically what they discovered. And when I'm talking about it, hopefully that'll be more apparent. The history of microbiology largely early concerned the debate about spontaneous generation and the theory of biogenesis, and that is, could organisms spontaneously generate from, what do you want to call it, inanimate matter? They had to get certain ingredients together and the vital force coming into it to make the spontaneous generation of the organism, or did all organisms come from a pre-existing organism? And that is known as the theory of biogenesis. Any question about that? We'll also talk about the golden age of microbiology. That's the early microbiology, where whenever microbiologists discovered something, it was something new. And that, you don't need to know the year, largely happened from 1857 to 1914, known as the golden age of microbiology. All right, we'll talk more detail about this, but that's the golden age of microbiology, which really begins, oops, not too far, when microbiology began. And you'll notice we will talk about a few things that really began before microbiology. All right. So in the history of microbiology, it begins in ancient times. Greater than 3.5 billion years ago, we can find in the fossil record the first fossils of life. You can find the first fossils of ancestors of bacteria. And they first appeared greater than 3.5 billion years ago. And here we're actually looking at, let me blow that up. There you go. Some fossils, fossil prokaryotic cells, which were fossil ancestors to the bacteria. This is the only date you should need to memorize. 3.5 billion years ago or longer is the first time we saw evidence of life on the planet Earth. You don't need to know any other dates after that point in time, but the next record is that we have records of the Babylonians recording recipes for making beer. And the beer was made by microbes. So we have records of microbes being used in the year 4300 BC in Babylon to make beer. Now, the Babylonians did not know the microbes were making the beer, and they did not even know what microbes were because microscopes weren't invented then, so they couldn't see them. But we do have evidence that microbes were used a long time ago to make beer. And you don't need to know this date, but you should realize that before the Babylonians wrote recipes for making beer, that before that, you had to have a fossil record for evidence of life. And then in 600 AD, we have written records of the Mayans making a fermented beverage, which is cacao, which to us would be very similar to chocolate. And it was a liquid, but it wasn't milk chocolate because there's no milk in it. It would be similar to taking baker's chocolate and melting it in water and then drinking it. It wouldn't be very appetizing. but the Mayans did ferment this beverage using microbes that did the fermentation to make cacao or chocolate. And like I said, before this happened, we had the Babylonians first writing down the beer recipes. And then before that, the first fossil record of life, meaning it is important that you should know the relative rank of things. Meaning before the Mayans could make chocolate, there had to be the Babylonians writing down the use of microbes to make beer. And then before that, there has to be the first fossil record of life. Any questions about any of that? As I stated, you do not need to know any other date other than this first date. All right. The next event in the history of microbiology was the invention of the microscope. In 1590, you don't need to know the date, Janssen invented the microscope. I don't know how he got the concept, but he put two lenses together and made a microscope. It was very primitive and didn't work very well, but in 1590, the first microscope was made. And this was actually before microbiology began, but we had to have the invention of the microscope before microbiology could begin, because you can't see a microbe without a microscope. The next important event in the history of microbiology was Robert Hooke used a microscope and saw the first living cells. Let me bring up a window of paint. I want to draw something out. There we go. Come back to the lesson now. So in 1665, you don't need to know this, the year, but Robert Hooke used a microscope and looked at something that was alive, and he saw cells. I guess I'll go to the picture now. Now, why he called them cells was because, where's my I'm not seeing the icons. Oh, here we go. I'm not a good artist, but we're going to try and do this anyways. He looked at shavings of cork under the microscope, and he saw cells like this, and he called them cells because it reminded him of a cell in a monastery. Oops, that's not very straight. We'll make that a straight line. And this would be the hallway in the monastery, and this would be the room that the monk slept in and lived in. And the monks called this a cell, and it reminded Robert Hooke of a cell in a monastery. And so he called the cells he was looking at in Cork a cell, because they looked like the cells in a monastery. And that's where the word cell comes from. Any questions about any of that? As I stated, Robert Hooke actually looked in shavings of cork. There we go. Which, if you recall, cork is the bark of the cork tree, So the cork tree has bark on it, which we peel off to get cork, and we use that cork to make cork that we use to cork a bottle. And those are three different ways that the word cork can be used. Any question about any of that? Anyways, that was the first time anyone had ever seen a cell. And that marked the beginning of the cell theory, which began in 1665 and went further. Virchow developed this theory much further, and he literally wrote it up, the cell theory, and published it. And we'll talk about it in a little bit. And he stated that all living things come from a cell and are composed of cells. And that was the cell theory, that all living things are composed of cells. Now, when Robert Hooke first saw cells in a plant cell, he found cells in plants, but he did not find cells in animals. And why do you think Robert Hooke did not find cells in animals? What do plants have that animal cells do not? And this made the cell much more visible under the primitive microscope available in 1665.
Cell walls.
Yeah, cells have a cell wall, and cell walls are very thick and they're easy to see. And the primitive microscopes were not very good and they were blurry. And it wasn't until, gosh, about 100 years later that the microscopes improved and we got good enough in focusing with the microscope that we could see cells in animals. And once we started seeing cells in all living animal tissue, as well as plant cells in all plant tissue, we started realizing that the cell theory was correct, that all living things are composed of cells. The above is not strictly microbiology, but it all needed to happen before microbiology could start, okay? Meaning that Robert Hooke was looking at plants, so that's not exactly a microbe. And then the development of the microscope was not specifically microbiology, but it needed to happen before microbiology could begin. As I stated, the early microbiology concerned the hypothesis about how life originated. There were two competing hypotheses to explain how life began. The first was the theory of spontaneous generation, and that is living things arose from non-living matter, and a vital force was needed to come into the non-living matter to make life, and then life would spontaneously generate. That was the first theory about where life came from. The second theory was biogenesis, that life only arises from other life. For example, everybody knows a baby only comes from its parents. So the baby comes from living organisms. Over a century ago, beliefs still included things like you threw a log into the fire, sometimes a newt would come out. And people would say, that's proof that newts spontaneously generate. You put wood in the fire and the wood and together, as long as the what is it? The vital force comes into it, spontaneously generated the nuke. And then people saw flies emerging from manure, and they'd say, the vital force comes into the manure and maybe out of the air. I don't know what else was there. And the manure come together to spontaneously generate the flies. And then maggots arising from a decaying corpse, people use that as evidence of spontaneous generation. Most were logical, none were scientific. Some were even amusing. Somebody wrote early, you can read about it in the next slide, that if you put sweaty underwear in a jar containing wheat grains in it, that mice would spontaneously generate. Okay, there we got the debate of spontaneous generation. I'm not going to cover that. Let me go back to this one here. What happened was the mice climbed into the jar to get the grains of wheat, and then they couldn't climb out of the jar because of the sides of the jar. And so the mice were trapped in the jar, and then people saw the mice there, and they probably noticed that some of the wheat was missing and the sweaty underwear. No idea what that was important, but somebody did that and stated that mice spontaneously generated. And like I stated, the mice just climbed into the jar to get the wheat and then couldn't climb out. They did not spontaneously generate. Let's take a brief sidetrack about this. It's important for talking about spontaneously generation. When we have a test tube with media in it and it's clear, that means there's no microbes growing in it. But if it's cloudy, and that's not very clear, let me see if I can blow that up even further. There, maybe you can see that's cloudy. It's because there are microbes growing in it, causing it to be turbulent or cloudy, as opposed to that, which is clear, at least right here. This is important because when we're talking about the spontaneous generation of microbes, people notice that when you had a tube filled with media, microbes would grow in it. You'd see growth. And they'd say, microbes spontaneously generated. The first debate about spontaneous generation was actually proved by Francesco Redi. You don't need to know the date, but Francesco Redi disproved the theory of spontaneous generation. And this was also the first time that a controlled scientific experiment was ever written up and reported in literature. So this is the first time a scientific experiment occurred by Francesco Redi, and he disproved the theory of spontaneous generation. What he did was he took 3 jars and completely sealed the jars having meat inside them, or he had 3 jars with meat inside that were open, and he had 3 jars that were completely covered with a fine wire mesh, and there was meat inside. Okay. In the jar that was completely open, maggots arose in the meat. In the jars that were completely sealed, there were no maggots. But people said, you completely sealed the jar, so the vital force could not get into the meat and cause the maggots to spontaneously generate. If you'd left the jar open, the vital force could have gotten in and there would have been maggots, is what they argued. So that's why he took 3 jars and covered it with a fine wire net. And then that prevented the maggots from arising in the meats. And this is the first time that somebody disproved the theory of spontaneous generation. After Francesco Redi's day, everyone agreed that large organisms like a maggot or larger could not spontaneously generate. Okay. However, people argued that microbes could spontaneously generate because if you had a tube, and even if you sealed the tube, microbes could grow in the tube. We'll talk a little bit further about that. But let's go back to the history of microbiology. The history of microbiology began with the first observations. And the first observations of microbes were done by Antony van Leeuwenhoek. I'm not Dutch, so I could be butchering his name. He wrote about his microbes that he saw in microscopes that he actually made himself. And at his day, he was the best lens grinder. He actually made his own lenses and then made his own microscopes. And he was the best lens grinder and microscope builder of his day. And he used his microscopes to see the first microbes and then wrote about it. And he described living microorganisms and almost everything he looked at. Microorganisms and scrapings from his teeth, in rainwater, in peppercorn infusions. And he also looked at water from the River Thames, the water that was used by the citizens of London to get their drinking water. And he found microbes in all of it. Any question about any of that? As I stated, he made his own microscopes, and his microscopes are very simple. They only had one magnification lens, so it was a simple microscope, not a compound microscope. But he was such a good lens grinder that his microscopes could see things that other people could not see with the microscopes of their days. And his microscopes were so good that he actually saw cells that were so small that they were not seen again by other microbiologists for nearly 100 years. Here's a drawing of some of the cells that he saw under his microscope. And if you recognize these, he's drawing bacteria. These are rods, these are cocci, these are rods. That's the movement of the rod, and that's a sporillum. Different bacteria. These cells were so small that they were not seen again by other microbiologists for 100 years. They needed the development of the microscopes to improve so that people could focus in and then see the bacteria that Antony van Leeuwenhoek, with his microscope, saw 100 years earlier. Any question about that? All right. So returning to the theory of biogenesis, Rudolf Virchow proposed the cell theory. You don't need to know in 1858, but he wrote up the theory that life only arises from pre-existing living cells. And this theory had been around for many years before he wrote it up, but he's the first one who wrote it up. And he wrote it up as the theory of biogenesis, that all living things come from pre-existing cells. And the argument continued for a few years about whether or not microbes could spontaneously generate. And that argument wasn't settled until 1861. Once again, you don't need to know the year, but it was settled by Louis Pasteur, who disproved the theory of spontaneous generation and thereby confirmed the theory of biogenesis, that all living cells come from pre-existing cells. Louis Pasteur did a number of things to disprove that spontaneous generation happens. First, he showed that microorganisms are present in the air. And so if you make a tube and then put media in it, and then even if you cap it, the microbes can fall into the tube from the air, and then they can grow in the tube. So they did not necessarily spontaneously generate, they fell down from the air. He also showed that microbes can be destroyed by heat. And so if you heat the tube and seal it, then the microbes would not grow in the tube. And then he disproved the theory of spontaneous generation by his famous swan neck flask experiments. What he did was he got a glass flask like this with a long glass neck, and then he poured media into it, and then he heated the glass part of the neck right there and bent it into what they call the swan neck. And then he heated the media to kill the microbes which were in the media or in the container. Okay, or they fell down from the air. And then he left this tube open to the air, so the vital force could get in to the media, but he saw no turbidity. There was no growth of the microbes in the swan neck. However, he could get growth in the tube if he took the media and he put it down to here and then brought the media back. What he showed was that microbes could fall down from the air, but then they're collected in this portion of the tube. They do not go up by gravity and then fall down into the media, unless you take the media down to here. So this disproved the theory of spontaneous generation. Any question about any of that? After Pasteur's day, nobody argued that microbes could spontaneously generate. Any question about any of that? All right, let me check. I think we're getting close to the time when the class is supposed to. And actually, let me put it on my thumb drive on another computer. That'll be quicker. Sorry, new term. Anyone know when the class is supposed to end? I'll get it here in a minute.
I believe 6:50.
Okay, if it's 6:50, then I'm gonna stop right here and continue. Let me just recheck that. I need to get this syllabus on my computer that I do the lesson from. Oh, come on. Yes, all right. So let's end the class here and I will see you in the lab at 7 o'clock.
I have a quick question.
Any questions?
Yes. So when you are going to upload the recordings onto YouTube, will that be at the end of the week or will it be after the class is over?
I uploaded, well, maybe after the lab, but I have uploaded the first lecture.
Okay.
Is that what you're asking?
Yes, I just wanted to know when it would be uploaded. I haven't checked recently, but I just wanted to ask first.
But yeah, it's probably not until after the lab, and the stuff does take time, meaning it has to finalize the program on my computer, and then I upload it to YouTube, and we put what is it called? The subscripts up so that you can go and read what's actually stated, and that takes time as well as the time for the process on YouTube. So it doesn't happen immediately, but usually about a half an hour after I upload it onto YouTube, you can then see the lesson on YouTube.
Okay, perfect.
Okay.
And then for the worksheet, the 001, do you want us to turn it in under week 1 lab?
You mean there's no link for week 00? Lab 00?
There was only week 1 from my page, unless I'm looking at the wrong place.
All right, let me look into this. There should be a lab 00. Where isn't that going? There we go. I know you can't see this, but I'm trying to get to Canvas. Let me share the screen here. Come on. My computer is making the oh, it's only one link. That's the problem. So under worksheet for lab 00 and lab 01, there are 2 documents.
Yes.
I'm pretty sure you can submit multiple documents. I don't have the link, but down here, there should be a submit button.
Okay.
Okay.
If it doesn't allow us to submit multiple documents,
I'm
if it only
pretty
allows-
sure it allow you to submit multiple documents.
Okay, sounds good. Thank you.
All right. All right, if there's no further questions, I'm going to end this.